I don't know about the rest of you, but I'm totally impressed with our trainees. I couldn't really tell without checking the bios who was a master's student, who was a postdoc, and that is not because our postdocs are underperforming. Those were really exceptional projects with lots of potential, translational potential, discovery potential, and I'm just delighted. I mean, also, I want to emphasize that they've had about 8 months worth of funding. Obviously, they've been training for longer in some cases, but delighted with the first Education Fund recipients. So congratulations to all of them, and look forward to seeing those publications and wanting the Rogers Centre to be kept informed of how your work is coming along. So it's my pleasure now to introduce the final session of the symposium. And here I'll preface the session by saying that we announced the Innovation Fund winners literally last week. So this is not about them showing data, although some of them may have preliminary data to show. It's really more about giving our community an opportunity to see what is getting funded and what we look forward to hearing about later. With permission of Dr. Honjo, I'm going to change the sequence because Ian Scott needs to go to his son's birthday party. So I'm going to start with Anthony Gramolini and Ian Scott to tell us about their seed funding project, functional analysis of novel regulators of cardiac function using the zebrafish model. We've had, I think, a little bit of a preview of how these two labs are coming together, and we look forward to hearing how they're going to spend our money.
Yeah, we clearly said we can spend money. Yeah.
I'll just do calisthenics. Need the crowd in calisthenics while Tony talks.
Yeah. As you heard, Mansour just gave indication that the notice for these awards just actually really was last week. But I think Ian and I both are here together. This is a project that actually had been in development probably for about 2 years, and we've been doing these kinds of things. The Rogers, this TRCRHR. We got in trouble for the Rogers Center. Yeah. So this actually, though, formalized what I think was one of these things that was percolating, and so it turned out to be a perfect union, I think, of two labs in complementary approaches. So we've had 2 extremely packed days of genetics, proteomics, squishing, stimulating stem cells, the whole gambit. But I think from our perspective, clearly we're biased, do we actually know all the proteins involved? We actually came into this project, do we know all the proteins involved that are in heart function and then are perturbed in heart disease? So this was our sort of our landing point, where my interests really were. We've been talking on and off for a few years with Ian, and that was our entry point. And then are any of these proteins we'd been doing, I think you guys had heard, some of our trainees and my own talk, was really going after things at the cell surface, and again, particularly in the cardiomyocyte. So again, you've been hearing everybody trying to think about drug repurposing, drug identification. We had an interest in that as well, and we were looking that if we could identify any of these molecules that theoretically are accessible, theoretically they're druggable. We can manipulate them, all of our informatics could identify those things. So this is where our big picture was. So we'd been on committees before, we'd actually been talking before. The data I think you've seen, we actually have some at least sort of rationale of how we all got here, and the pipeline of identifying some of these proteins. But this actually was, I think, a bona fide real integration of one of these multi-platform, multi-institute, and I can let Ian talk about, I think, the beauty. I don't have Rodrigo. I know we've joked at the University of Toronto, but over the last few years, I've become an extreme champion and a proponent of the lower organism models that actually, when they do have convergence, it actually allows fast screening, direct phenotype analysis. And that's how we really ended up walking into Ian's office and said, well, I have a couple of things I think we bring some value. how do we go ahead? And then Ian came on, and this is where we are right now.
Excellent. Sure. Thanks, Tony. Nice segue. So yes, good afternoon, everybody. So really, my lab has really spent the last several years studying cardiovascular development in disease in zebrafish. We have a lot of interest in early heart development, congenital heart disease, and then, as we've heard quite a bit over the last few days, cardiac regeneration as well, which is a very interesting property zebrafish have. So we use this model for a number of reasons. As Tony mentioned, one is we can do functional analysis, certainly of heart development, very readily. So this is a movie of a 2-day-old zebrafish heart. So the embryos are optically transparent, externally laid, so we can really watch all states of heart development happening in real time. And we can really, at a high level of resolution, under a confocal microscope, look at all parameters of biology, basically cell numbers, cell shape. Along with Tony, we can look at physiology now, as well as calcium handling, heart rate, all these different parameters of development that we're interested in. So this has been the model, and as Tony mentioned, the last few years we've actually, with some success, and actually published success, actually, which has been fantastic. We heard about some of that really nice work this afternoon in one of the trainee talks. Been using the fish model sort of in the background, kind of stealing money from projects here and there to do it. Doing it mostly with morpholinos to knock down genes. So anyone who knows anything about zebrafish knows that morpholinos to fish people are kind of like the Trump presidential candidacy to Americans. Some people support it, and a lot of people would really like to see some other options instead. That would be nice. And so with this funding now, with this dedicated funding, we're really excited now to go forward and actually make the proper CRISPR mutants, which we've really scaled up in our lab in the last few years, and actually study this in much greater detail. Here's the giant pipeline slide, which I'll let Tony get through, since this is his baby.
So, as Mansour indicated, what he wanted us really here was to talk about what is our vision now for the year. From at least a lot of our data, I think we've made it clear we've been doing lots of mass spectrometry. We have pretty much data overload. And internally, we've been looking at that. You've seen instances of sort of these top 1 candidate or the number 2 candidate. But we actually said, well, if we took not that risk-averse approach, but really went in and said, could we go after a much greater number of candidates? Could we actually make some real dent? That's the beauty of the fish. So we could never afford a mouse or 10 mice to make transgenics or knockouts, but we can do it in the fish. And so we proposed, right? So in this grant, this is now like 2 months ago, I think, when we actually put this grant formally in. We had said we're going to take all this data, we're going to look at cardiomyocyte specificity, we're going to go after very unique things, integrate it all, and come back. We actually proposed in the grant itself 10 of these extremely novel, cardiomyocyte-enriched, unstudied molecules that we felt would have a direct implication in cardiac function and or development. So we pitched this idea that we actually have with Ian an ability to make these lines and then immediately assess them. And then we should have been able to go through these 10 within this year with a comprehensive approach, dedicated people on this. But I think even as we went through the meeting, we added, and we need to be very clear, this was our original set of studies. We're going to talk about our first one, because clearly we're good to go with one. But I think it's a perfect opportunity. I think SEMA stepped out and some of the sick kids people stepped out. But as we go through, again, Ian and I on a one-off of talk, this is a good idea. We should talk to SEMA. We should look at some of this sequencing data. We actually should formalize, integrate some of this work across the platform. And so I think what we have to do is, as SEMA's data sets are getting stronger and better, and actually I think we both even have a SHOX paper which has been in review for a bit, unfortunately. That's a good premise, that we actually have some degree of identification in people, in pediatric, these are tetralogy patients or early congenital myopathy patients. Can we actually integrate that? So we have to do that. So I think this list may jiggle a little bit, but this is the idea to actually, I think, bring the power and the thinking of the Rogers Center. But the idea is to make these fish, get going, make the correct mutants, and then, as Ian said, look for, we lost the beautiful movie, but look for cardiac development, function, biochemistry, physiology, all coming together. And so, why do we actually feel this is going to work? It might not. Being honest, it might not work. But we have precedents. So that TM65 paper, that number 1 candidate of our plasma membrane, highly conserved, we actually made that fish with Ian. And in fact, because of that radical high extent of evolution conservation, our reagents worked, the specificity, that red heart, if we look here, our antibody worked in the fish, which is amazing. And in fact, that's endogenous protein in the zebrafish heart that's only in the heart. So in fact, these precedent is set, the proof of principle that I think it could work. Here, that again, I've laughed with Ian and maybe at myself. That's a happy fish. These are all dead fish. I can interpret that this is absolutely important. And I think that's the extent that we're not looking for nuanced differences. We're looking for real central protein regulatory markers. And so we come back. So that was our protein that we just published about 6 months ago. We're now looking the number 2 candidate. I think you heard Frank talk about. Also, just I think because of our MS identification led to these proteins being highly conserved between at least upper human down to the mouse. There is conservation. Our other protein here is about 80% conserved to fish. So I think that this is the idea. And in fact, in Frank's talk, he had this data that, again, Ian went and had another morpholino, and not as severe as TM65, but we actually have congenital development problems at least in this fish, in cardiac looping and early stages of cardiac development, cardiac function being impaired. And as we follow this, we're going after really how all these proteins, one of the things that I think we can do is find a function. How do they work? What do they bind? Are they affected? We've done the overlay with heart failure and disease, so hopefully these proteins continue to show up in heart failure and pathophysiology. But this is the premise that I think there's precedence that it's working. So the idea was to go rationalize these 10, make some mutant mice, sorry, Ian. No. Make these fish, which are very fast, very effective. But I think now, actually, because of this meeting, we should really reflect, probably in the next couple of weeks, reprioritize our candidate list, and then continue to go full steam ahead with making these fish, doing the developmental biology, the physiology as we go forward. And we did this the other night. That I think this was 1 year, but we are hoping for a second year of funding. And but I think if this actually has some success and we can move forward and show the utility of all the things that I think are mandatory in this program, that we could envision additional mutations. We could envision, even if things work out, identified human mutations in some of these genes, not just an all-out knockout, but begin to actually use some modeling system. And that's where we go. Ian has actually done some drug screening in fish. I think you've seen some of the stem cell at least work. The fish are, I think, just as good, maybe different, but just as good as the human stem cell work for at least some of these initial screens. They're a full functioning organ. So, I mean, that will be, I think, the way that we have to think about year 1. And then, well, everybody has to worry about sustainability. So what will we do in year 2? How will we continue to move these projects forward? So I think that sums
it up pretty nicely. Yes, that's it.
Questions, and maybe I'll start, if I may. So we, the review, again, this went through a phase of letter of intent. We sent this out to reviewers really internationally, and it's one of the very highly rated grants. Everyone likes your idea. But you're saying you're going to pick 10. You may refine the 10 based on what's emerging from CIMA's data but your hypothesis is that these are enriched in the heart, right? So, but there are lots of people have been here with their own gene or their own pathway. I've heard some other mouse models. Phyllis has that beautiful mule thing. So what I'm wondering is should there be a core to allow zebrafish cardiomyocyte? I guess I'm playing devil's advocate. How are you going to do just 10 fish? You're going to hire one person for 1 year? What's the What's the operational?
Yeah, that's a great question, Mansour. So I mean, part of the Sick Kids arm of the Rogers Center does involve a zebrafish modeling arm. So as the genetic studies ramp up, and obviously an enormous amount of new candidate genes will be made, there will be a modeling aspect to that. There is a limit to how many mutants we can make in the lab. I can make a plug for zebrafish. The Sick Kids also has a zebrafish core facility, which is now making CRISPR mutants at basically a cost recovery basis for members of the community. So certainly other members of the Rogers Center, if they have really interesting genes that they can either talk to us, we're always happy to talk to people, that could go onto our list, and or people could generate those mutants through the core and then house them in my lab.
So practical question, what's it going to cost and how fast can they get it made?
Right, so the current pricing in the core facility is somewhere in the order of $3,000 for a mutant, I believe.
Okay.
It's about a 6-month process to get stable carriers, and then the initial cardiac analysis in our lab is very quick. The initial phenotyping at least is very fast, and then obviously it gets more detailed.
Okay. Michelle. You can't hear without that. Yeah.
I was just going to ask sort of the same. Whoops. Very enthused. The same line of questions, but I just wanted to know for you guys, how long did it take you to get the data that you presented beautifully in the paper with the TMEM mutant zebrafish?
And the REAP.
Yeah.
And the REAP. Yeah, and the REAP.
The initial fish data, this is all done as morpholino, so that's a caveat. So the initial morpholino data is quite quick. Because we've done a lot of this work, we know how to properly control for morpholino experiments. I should say that about half of published morpholino studies are wrong. It's just toxicity effects. But we have a lot of experience studying this. We use multiple morpholinos, et cetera. So I think the now was the Cas9 technology. Making the mutants is really going to be the way to go. This is really going to allow us to definitively show, do these mutants really have an effect on the cardiovascular system? Excitingly now, knock-in technologies are becoming feasible. So as the sequencing data gets ramped up, we could start thinking about not just making total loss of function mutants, which is not what we often see in patients, but actually making human-specific point mutations, assuming those residues are conserved in fish, which for at least the proteins we've been studying, and that is one of our prioritizations, is how conserved are these proteins, is going to be feasible.
So I have another relatively naive question. The zebrafish as an exciting model has been around for a long time, right? And people have even done sort of like genome-wide screening of knockout. Yeah, right. So is there a publicly available database that you can go in without making the fish to sort of say, that mutant was made 10 years ago in Fishman's lab at the MGH, and it's matching with one of your top 10, that you wouldn't make it?
Sure. So there is a website called zfin.org or zfin.org, depending what your nationality. And it actually curates the entire published zebrafish research ohm, if you will. So you type in a gene name, it'll tell you everything that's ever been published on that gene. So expression data, are there loss of function studies, what papers have been published on this, that information is all there.
So I'm assuming you guys have gone there first?
Yes, of course, yes.
And so can you give us a sense, how robust is ZFIN or whatever you call it? Is it got 1,000 mutants in there, 100 mutants?
I would have to wager it would be in sort of the hundreds approaching the thousands, I would think. Right. Not all cardiac centric. Several mutants were recovered. One of the initial screens was done by Fishman, was obviously cardiovascular centric. Yeah. They didn't isolate all those mutants as far as they made mutants, but they didn't. Yeah. This was old school positional cloning days. We were doing back walking and things that all the trainees now wouldn't understand anymore. So they didn't clone all those mutants. So that's sort of the short answer.
Got it. Sarah.
Is that on? Yes. So just to ask you how to start to think about translating this clinically. Obviously, finding new genes is fantastic, but one of the issues that we deal with are the variants of uncertain significance in old genes or known genes. Can you see that there's going to be a high throughput facility available for us when we get these VUSs back, where we would love to know whether that's the pathogenic mutation in the family, but we just don't have enough segregation or just not enough literature information? Would there be, I know it's quite boring, but on the other hand, that's what the translation is going to have to look like.
Sure.
I don't think it's boring. I think it's very exciting.
No, it's not at all. I think novelty is always exciting, right? Yeah. So I certainly wouldn't call it a high throughput way to analyze things. I would call it a low to mid, maybe a lid throughput kind of approach. So, making these knock-in, especially knock-in mutants, so very specific point mutations, is a little more laborious. These techniques I mean, you can open an issue of Science or Nature almost every week, and new CRISPR techniques are approaching, which are getting much more efficient. Basically, was every month that we do it. So, so the throughput is not going to be massive, but we can certainly. It's going to have to be a case-by-case basis, I think, of prioritizing what are the really interesting things. I mean, I think the exciting thing about making the genetic mutants instead of the morphins, for example, is we can start looking at genetic interactions between different mutants. So instead of looking at just completely wiping out protein function, do the heterozygotes have more subtle adult phenotypes? Do compound heterozygotes have phenotypes? I think that's where I really see the fish work going that could be really quite interesting, as well as the small molecule screening, of course.
Just
Yes, Sarah.
a quick follow-up question then. If it's not going to be zebrafish to give us the high throughput, do you think that there is an alternative on the horizon, or are we going to be stuck with paying sort of $3,000 to work out whether the thing we've already paid $1,000 for is actually the pathogenic variant?
I think that's again. Certainly the cells, although obviously working with iPS cells is not exactly cheap either. But I think, as we've heard from several of the talks, it's really going to depend on what you want to study, right? I mean, if we're looking at long QT type of phenotypes, then the iPS cell model is really proven to be an incredibly robust model for that. If you're looking at tetralogy of Fallot or septation defects, then the state of the art right now for the in vitro cultures is not there yet, although it might very well get there at some point. I mean, we saw some really nice work today in that regards, right? But so I think it really depends on the nature of the phenotype. I don't want to shoehorn everything into one case, because I don't think that's going to be the case.
It's a really good question, Sarah. One that I think that as a center we should maybe strategically try and answer. The costs of the IPS stuff are actually pretty high.
Yes, for sure.
Very high. Manufacturing, assembly, notwithstanding what Melitza can do three-dimensionally. It's pretty impressive. I'm going to ask a really dumb question, because I always like to tell the students there are no dumb questions. You showed the conservation to fish, right? Vertebrate, it's all vertebrate. But we have Rodrigo with the Drosophila. And I remember, because my first research project as a summer student was Drosophila. There are some mutations in the Drosophila that map right up through. Do either of you, I don't see Rodrigo here.
He was here earlier. I talked to Rodrigo a bit earlier on today.
Yeah.
I'm not sure if he's still here. I mean, the classic example is Tin Man, right? And KX 2.5 was found in fly, and that's like still the hallmark marker for cardiac progenitors.
Yeah.
Mutated in children with congenital heart defects. So, I was actually talking to Rodrigo a bit about this morning, because he's started to do some of this work in his lab now too. But so Rolf Baldmer at UCSD has really been the pioneer in this. They've done a lot of work studying the fly dorsal vessel or the heart, and a lot of the ion channels are conserved.
They are. Yeah.
They do a lot of the pharmacology. They take the drugs that affect human cardiomyocytes, and they have very similar effects on the fly.
Yeah.
So there's that is perhaps a more high throughput model one could consider. Also, that's nice. I mean, this is the beauty of the centers. We should think about combining all the different tools that we have for this purpose, right?
Yes, Slava.
Yeah. So just to follow up on your question, Mansoor, I was going to talk about the fruit fly. So obviously, every gene in a fruit fly has been knocked out, and it's been knocked out in a cardiac-specific fashion, and you can deliver cardiac-specific stress. So Joseph Penninger did this in his fly screen for heart, and they had a lot of hits, and many of those hits were already known proteins, and they published a novel paper in Cell about a mutant. What they did is exactly what you're proposing, Tony. They took genome-wide association data that was subgenome-wide significant, so in the noise, and link that to their hits in Drosophila to focus in on novel variants, essentially. So these things, there weren't enough people, especially in heart failure, we may not enroll, we were hearing earlier, we may not enroll enough people to ever really do great studies like that, but there are fly libraries around, some of which can be refined in the fish, and they can be proven in the mouse, and then there's many human data sets that can be linked to this.
Yeah,
That's an excellent point.
Right.
Thanks. Yeah.
excellent point. Thank you, Slava. So we'll make sure we get Rodrigo next year to talk about that. Yeah. Anyone else? All right, thanks, guys.
you.
Good luck.
Thank you.